Namaste students, I am Jibin Dr. Aziz Sumit Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic downstream processing step number 2. So first discuss about the downstream processing. What is the downstream processing? Downstream processing the various stages of the processing that occur after the fermentation process. After completion of the fermentation process or bioconversion stage, which includes separation, purification, packaging of the product so that it can be sent to the market. So this is the general introduction. So in the downstream processing, various steps are involved, which include the removal of insolubles, product isolation, product purification, product polishing, and finally it is sent to the market. In this video, I'm going to discuss about the second step, which is the product isolation. In previous video, I discussed about the removal of the insoluble particles, means the particles which are insoluble and can be easily separated. But product isolation is little bit more hard step. So product isolation, removal of those components whose properties vary markedly from that of the desired product. So they need to be separated. So for example, water is the chief impurity, and isolation steps are designed to remove water by the dialysis process, reducing the volume of the water, concentrating the product, liquid extraction, adsorption, ultrafiltration, precipitation are some of the unit operations which are involved in the product isolation. So first step is liquid extraction, liquid extraction. So it is the example of the separation of the liquid particles from the liquid. Okay, it is a separation process that takes the advantage of the relative solubilities of the soluble in the immiscible solvent. Means they are the particles which need to be separated are both are the uh, liquid in nature. So the separation will be conducted on the basis of the solubility in different solvent. Out of those two particles, one particle is not soluble in the solvent, and one particle is soluble in the one solvent. So the soluble particle will be easily separated. Solute is dissolved more readily and become more concentrated in the solvent in which it has a high solubility. A partial separation occurs when a number of solutes have different relative solubilities in the two solvents used. Solvents should be non-toxic, selective, inexpensive, and immiscible with growth, and should have a high distribution coefficient of the product. So these must be the properties of the solute. What must be the properties? The solvent must be non-toxic so that it cannot disturb the property of the original product. It must be selective because it must be selective because it need to be added in the media so that it cannot disturb the another particles. It will not dissolve another particles. Okay, only one part, one liquids will be separated. So this must be a property of this solvent. Next, inexpensive. The solvent should be inexpensive so that the total cost of the product may not raise, and it should be highly distributed. It must be easily available. So these are the properties of the solvents. Now adsorption. Adsorption is also a method for the isolation. Sorry, product isolation. Adsorption is the surface phenomenon. Means it is a binding of the molecules to the surface and different from the absorption. Means absorption and adsorption are the two different terms. So it is a binding molecule to surface. Okay, the binding of the substrate is weak and reversible. Means if a substance has been bound and we have added a adsorbent, the adsorbent binding to the particular molecule is weak and sometimes it may also dissociate. Okay. The compounds containing the chromogenic group are usually strongly absorbed on the activated carbon. So activated carbon is a group adsorbent. The common adsorbent used are activated carbon, silica gel, alumina. So these are the examples of the adsorbents which will adsorb the particle on the surface and it will be easily separated. Okay, and why they are good, good adsorbent? Because they are present in normal surface areas per unit phase. Next, the ultrafiltration. Ultrafiltration is a good method for the production for the product isolation. Ultrafiltration is basically a pressure driven separation process. The operating pressure is usually between 0.1 to 1 million. So ultrafiltration is governed by a scanning principle and it is dependent on the particle size. Like membrane separation, it is also a method for the separation of the particles by membrane separation method. Ultrafiltration membrane are different from the normal membranes, and the ultrafiltration membrane have a pore size between the 1 nanometer to 100 nanometer. Means they are in the diameter of 10 to 2000 Armstrong and have allowing tension of the compounds with the molecular weight of 300 to the 5 less Daltons. So typically this ultrafiltration process is suitable for the retaining biomolecules, bacteria, viruses, polymers, colloidal, some sugar molecules. So these molecules are separated by ultrafiltration and it is a simple method and bacteria will be easily separated, viruses will be easily separated by the process of the ultrafiltration. It is a simple method and mostly some ultrafiltration is also used in the drinking water also. Now for precipitation. What is the precipitation? Precipitation is the formation of solid in a solution using a chemical reaction. Solid formed is called the precipitate. What is the solid precipitation? Suppose there are three tubes. Okay, there are three tubes. Uh, the three tubes contain some mixture. Okay? They have some solution, and from that solution there are some suspension. So we are adding some chemicals to the suspension, then the sum of the particles will get attached to each other and they will sediment on the bottom. Okay, so solid formed is called as the precipitate. So that's all the solid particles which are present in the suspension will be settled down, and this solid particle separation process is known as the precipitate, and the liquid remaining above the solution is known as the supernatant. Precipitation is used by some salts such as ammonium and sodium sulfate. They are used for the separation of proteins. to precipitate. The organic solvent methanol are also used to precipitate, and they are used for the separation of the dextrins. Chilled ethanol is used, and acetone is also used for the protein precipitation, for the separation of the protein. Non-ionic polymers such as the polyethylene glycol is used in precipitation. So these all are the examples of the precipitation reactions. So I hope that the students will be better understand what is the precipitation reaction and other examples which are described in this lecture. It's such a liquid extraction, adsorption, ultrafiltration, precipitation. Okay. These I, I explained it in context of the students. I hope that students will be better understand it. So what kind of questions which can be asked in this lecture? The question which can be asked in this topic is that explain the second step of the downstream process, second stage of the downstream processing, and explain different stages, name of the different stages which are involved in the downstream process. So I hope that it will be helpful for the students. So my lecture is over here. Thank you very much.